This method is designed to answer key questions in the field of inorganic biochemistry about how to maintain enzyme activity in proteins that contain oxygen-sensitive metals.
The main advantage of this technique is that it helps you visualize the ways in which you can maintain a protein's metal in its reduced state.
Generally, individuals new to this technique will struggle because it is difficult to maintain materials and reagents to be completely anaerobic.
24 hours after inducing protein expression, collect the cells by centrifugation and resuspend the bacterial cell pellet in 20 milliliters of amylose column buffer per 1.5 liters of growth. Add 1 milligram of lysozyme to the cell suspension and stir the cells for 20 minutes on ice. At the end of the incubation, lyse the resuspended cell solution via high-pressure homogenization with 3 to 5 passes at approximately 15,000 psi and transfer the protein to a 50 milliliter centrifuge tube. Flush the head space with nitrogen for 1 minute and centrifuge the cell lysate to remove the insoluble debris. Transfer the supernatant to at least one 50 milliliter tube and cap the lysate with a rubber septum. Using a 20-gauge needle, slowly bubble nitrogen gas through the solution for 2 minutes to displace the oxygen. Then wrap the septum with paraffin film, secure the parafilm with copper wire, and place the supernatant into the glove box with the column materials. When the solvent is just above the resin bed in a previously prepared amylose column, load 50